Welcome to Brain Atlas. Chapter 2, Senses. So first we'll be talking about the vision. 30% of our cerebral cortex controls vision, and the first study organism was a Drosophila to learn how light is converted into electricity in our brains. The light enters our eye through many parts. First is the cornea, which is the outer cover of the eye. Next is the pupil, which is the hole that takes in the light. The iris controls the size of the pupil, determining whether we need a lot of light or less amount of light to detect what the image is. The retina is right behind the lens, and the lens bends the light onto the cell layer called the retina. The retina is actually made up of 3 cell layers, which we will talk about. The optic nerve basically sends signals from the retina to the brain, especially the occipital lobe, where the signals are processed. Retina has 3 layers. Photoreceptors come in 2 types, rods and cones. 90% of our photoreceptors are rods and only 5% are cones. Rods come in black and white. Basically, it helps us see in black and white, and they are really sensitive. But cones let us see in fine detail and incorporate the colors red, green, and blue. Combinations of these colors are what let us see different types of colors over the spectrum. We have 125 million of these in each eye. Interneuron is basically the processor of light and the relayer of info to the photoreceptors. Ganglion cells do the same thing except to the interneurons, and they also have long axons that exit the retina to form the optic nerve. Some eye info. The cornea is the outermost layer sheet. Sclera is the white outer layer of the eyeball. Iris regulates how much light enters. The pupil regulates the size of the iris regulates the size of the pupil. The pupil regulates how much light enters. Lens flips the image. Fovea is the center of the retina, highly populated by cones. Macula around the fovea is crucial for driving and reading. And the blind spot, also called the optic disc, is the only part of the retina which doesn't have photoreceptors. Thus, light landing on the blind spot is not visible to us. So there's a correlation between the eye and the brain. So the image goes into our eyes, and when it passes the cornea, sclera, iris, and pupil, when it reaches the lens, it turns upside down and reaches the retina through the aqueous humor. And then this is called binocular vision because we are seeing with both of our eyes. The ganglion cells on the retina collect the image info and map the scene. The left eye's image half of it goes to the left side of the brain and half of it goes to the right side of the brain. And then the right eye's image, half of it goes to the left optic nerve, half of it goes to the right optic nerve, and then after it reaches the brain, the parts of the image that go to the left side of the brain are the left halves of each of the eyes, and the right halves of each of the eyes go to the right side of the brain. Simply said, most of our right side's vision is processed in the left side of the brain, and most of our left eye's vision is processed in our right side of the brain. After passing through the lateral geniculate nucleus, it goes to the primary visual cortex, where the picture is flipped over and understood. There are 2 streams in the primary visual cortex. One is the ventral or what stream. This basically controls info like what is happening around you to make you recognize your parents or your dog or anything that is familiar to you. The dorsal controls your surroundings. We have to recognize what surroundings are around us, and so the dorsal or the where stream records what surroundings we see and if they are important or not, so we can remember it the next time. There are two main visual disorders that we have to know. Strabismus is basically the imperfectly aligned eye, and so the fusion between both eye images don't happen, and hence one eye gets favored and the other eye is covered by the patient because it helps them see better. But and then the eye that is covered loses its vision. And so this can be treated before the age of 4 or like as early as possible. Mostly before the age of 4. Macular degeneration is the degeneration of ganglion cells in the macula, and this is really important because this is the main part of the retina that receives the images. And so degeneration in this part would cause blindness, and this is the leading cause of blindness in people 55 plus aged. There's some main stuff about the ear we have to know. As seen on the right, the anatomy states that vibrations enter our ear as air pressure through the pinna. After reaching the pinna, it goes to the ear canal where it reaches the eardrum, or also called the tympanic membrane. Here, the membrane vibrates as the air pressure presses against it, and these vibrations are sent to the smaller bones connecting to the eardrum, which amplify the sound. These are called the hammer, incus, and stapes. They also have other names. The hammer is also called the malleus, incus is also called the anvil, and stapes is also called stirrup. The stapes pushes the oval window, and this groups up the fluid and pressure separately to send it through the cochlea. These vibrations and pressure on the hair cells in the cochlea are analyzed. The hair cells convert these mechanical vibrations into electrical signals to send it to the brain. So here's the inner ear magnified. Mechanical signals convert into electrical signals through a large process. A membrane called the basilar membrane covers the inside of the cochlea. This membrane is what measures the frequency of the sound by seeing the frequency at which sound is pushing the hair cells. The hair cells, called stereocilia, move right and left against the tectoral membrane, which then responds by triggering the release of ions. These ions are what turn the mechanical vibrations into electrical signals, and these signals are sent through the auditory nerve to the brain. Now, sound in the brain is processed in different ways. Each part of the brain processes a different aspect of sound. The thalamus and brainstem process the sound location, origin, and the direction where the sound is coming from. The auditory cortex and the temporal lobe keep the frequency map that was recorded by the basilar membrane. The auditory neurons record different frequencies and know how to deal with them. Cortical neurons record other characteristics of sound like intensity, duration of sound, and rhythm of sound. The auditory cortex is basically the data collector. It collects all the data from the different parts of the brain that process sound, and it identifies if it knows the sound or not. Basically, it processes and identifies if it's heard it before, maybe like a specific voice or an instrument, by using characteristics like rhythm, tone, frequency, intensity, duration, direction where it's coming from, and all of that. Auditory disorders include hearing loss and Wernicke's aphasia. Hearing loss is due to the loss of hair cells or stereocilia in the cochlea. Unfortunately, hair cells don't grow back, and so to regrow these hair cells, neurogenesis is being researched for a possibility for this. Wernicke's area is located in the auditory cortex, and it is responsible for making sense of sounds. A damage to this part is called the Wernicke's aphasia, where there's this inability to understand speech. Next, we'll move on to gustation. Gustation is the ability to taste. A big part of gustation is olfaction, which we will talk about in a few moments. Olfactory neurons are also the only sensory neurons that regrow. Our tongue has many small buds called taste buds, as seen on the right. We have 5,000 to 10,000 taste buds, and each of those taste buds has 50 to 100 sensory cells, and each of those sensory cells have a job. The job could include recording sweet, recording salt, recording sour, recording bitter, or recording savory taste. These are not the identifiers, but they just record the signs of these identifiers. And then these signals are sent to the brainstem through three main cranial nerves called facial, glossopharyngeal, and vagus nerves. Then it gets sent to the gustatory cortex and to the insula where taste is finally identified. Next, we'll get on to olfaction. Olfaction is the ability to smell, and smells enter through the nostril and attach to olfactory cells in the nasal cavity. The axons of these cells, like the ganglion cells, form a nerve that passes through the olfactory bulb. The info then goes to the olfactory cortex, where the information is processed to identify it. 1,000 types of olfactory cells exist, and so 20,000 types of smells can be identified. Olfactory cells are covered with cilia, like the ear, and these respond to the smell molecules instead of the sound molecules. Combinations of smells can also be identified, and it takes only 110 milliseconds to identify the smell. Touch is a different concept. It includes pain, temperature, texture, and a lot more. The biggest touch organ or sensory organ is our skin, which records touch. Touch receptors are in our skin, and these collect sensory signals to send to the spinal cord. In an emergency, the spinal cord responds immediately, like if you're touching fire or something really dangerous. If it can take time, like detecting what that thing is by touching a color pencil or a pencil, you know it is a pencil. And that takes time because the touch signal has to be sent to the somatosensory cortex to be recorded and identified. The signals then go through A beta fibers, go faster because they're myelinated, and signals that go through C fibers are slower due to the lack of myelin. Touch receptors are also more densely packed in sensitive areas like our fingertips, and they are not that densely packed or sparse in our insensitive areas like the back. Pain is a main concept in touch, and it is a sensory signal that can correlate with emotions. So this has two aspects, sensory and emotions. Pain is not damage itself, but a warning before damage. Nociceptors respond to danger by damaging tissues, so pain is felt. Common dangers are temperature, visible wounds, hidden wounds, and chemical effects like poisoning. Prostaglandins increase the feelings of pain more intensely or painful in cases of real emergencies. Neuropathic pain is a result of a malfunction in the nervous system when the nervous system is not behaving properly. So pain occurs even when there is no danger. Pain reaches the spinal cord through A delta and C fibers. C fibers are slower, so they cause less intense pain. The periaqueductal matter located in the brainstem controls pain. Endorphins are released by this, and these act like analgesic morphine, which is basically the changer of pain feelings. These basically reduce pain. And another way to decrease pain is stopping emotions by doing cognitive behavioral therapy. So in summary of pain, Two ways of decreasing pain is through opioids, which reduce pain, which is the sensory aspect of it. And another way is emotional aspect, which is cognitive behavioral therapy. Thank you for watching.